My bad, everybody. You'll be fine. You have the key info. I promise I won't scramble it so bad this time. Okay. Pathogens. Yeah, sorry. Pathogens are unique. They're shaped very oddly. Who's ever seen a picture of COVID? It's that ugly spherical looking thing. It does not look like a human cell at all. Same with bacteria. You have pieces of these pathogen that are distinct. Those are antigens. So the piece like a spike protein from COVID or an LPS receptor on bacteria, these are antigen. On antigen, there's another definition called epitope. It is an even more small piece of that protein gene. What your body is tasked with, if it is losing the fight to a number of viruses or a huge number of bacterium, is it must evolve a response to one of these pieces. It will take that piece and use that to say anything that looks like this will die. It takes time. So, first step in the adaptive response is actually from a couple cells that sort of bridge the gap between innate and adaptive. Monocytes or phagocytes, these are macrophages and dendritic cells. So in that initial fight that we saw, macrophages will go up and eat bacteria. That kills a lot of them. It's helpful. But the true key of what macrophages and dendritic cells are able to do is that both of these are able to not only eat and consume some of the offending pathogen, but what they will do is they will display that pathogen on their dendrites, or they will display it on top of their cell. It will display that piece that they ate. So when this macrophage eats this bacteria, it's going to take pieces of it and start putting it around itself, kind of coating itself in a foreign substance, right? That is an alert to say, whatever this is on me that I just ate, it's here. We need help. So I'm going to say rule number 2 is coded pathogen as a signal. Rule number 1 is obviously just take some down. So with rule number 2 in mind, if the battle is going very poorly against whatever you're against, these will take themselves, they will meet a T cell, and they will say, okay, I am coded in this antigen. And these cartoons are fun, sorry. I'm coded in this antigen. Here is a T cell. It is going to present this piece of pathogen that's winning the fight to a T cell and say, okay, like it's go time. It's time to evolve a big response now. This is bad. So from the battlefield, they'll eat those pieces. They will take them to our first adaptive person, a T cell. And a lot of you have probably heard of T cells before, right? Kill pathogen, stuff like that. They're only going to be triggered to do that if they are presented a piece. So macrophages and dendritic cells are sort of that link to the adaptive immunity side. I know I'm kind of blocking out some of that right there. This slide is very helpful. So of all the key info slides, this wouldn't be the worst thing for you to take into a PowerPoint and like sort of make little annotations to, right? Not the worst thing on the planet, not the worst study guide. Another fun thing, you can kind of take a little break here while you're looking at this, so I'll leave it here, is with the two-sided sheet of notes that you can bring to the test, you're totally welcome to print an image on this thing and bring it. That's okay. You can handwrite it too. But people were always like, oh no, am I breaking the rules? I was like, no, you can put pretty much anything on it besides like your buddy's cell phone number. I don't know how you get that past me, but yeah, we'll see. So in this case, this is also sort of our first compare contrast. Literally every immunology class will say, what's the difference between the innate and adaptive immune system? So everything we've met on the left, we're going to meet the two on the right. But when you actually get sick, it's about 3 to 7 days, right? B and T cells take about that long to evolve a response. It is slower, costs a lot of energy. That's why you go down. That's why you like get sick and you're tired. It's because all your little mitochondria are working in those cells. You shut down the other systems. The advantages of the innate immune system that we already saw. Very fast, quick, we're good. One thing that's very exciting that we get to see in the end, or like in the middle of class, that your adaptive immune system can remember something that it was presented years ago. Your innate immune system has a limited capacity for that. So, easy points. When I ask you, and I will, what is not a difference between these two systems, let's say? Or it would be more open-ended. Give me 3 distinct differences between the 3, or between the 2. Okay, so first off, here's where they come from. Thymus and spleen. Thymus, T cell. Spleen, B cell. Sorry, it didn't actually work out. And technically bone marrow for B cell too. That's not actually where the B comes from. We'll actually see. B cell response and T cell response. Yeah. There we go. So again, we didn't really know what the spleen was for like 150 years ago, so it was always like, take it out. That was a mistake. You still have a lot of lymph nodes running around. Remember those little lymph nodes like kind of running around all over the place with like kind of little hubs everywhere? You can still mount sort of a headquarters response without either organ, but it's not great. So these are the headquarters of both adaptive cells, the T and the Bs. Here's another key distinction. We'll kind of revisit this as well. And this is often a point of confusion for a lot of things. T cells attack infected cells. And what I mean by that is that when a virus goes inside one of your cells, it starts to kind of corrupt what that cell looks like. T cells are good at sniffing that out. B cells are much better at sending out what we'll meet in a sec there on the surface, an antibody, to go and kill things outside the cell. So I'm going to list some targets. Think of those pathogens that we met, parasites, protozoa, bacteria. All right, I think I need to replace this. So it's not like a strict golden rule. So for example, COVID is a virus. T cells can kill infected COVID cells. B cells can produce antibodies that kind of shower the free-floating virus. But for the most part, T cells don't have a huge role against parasites, bacteria as much sometimes, I'd say. Think about the big stuff outside the cells, B cells will attack. Time to meet. This is probably one of the more important things that we'll see throughout the class. So I think we'll do this slide and then we'll kind of take a break. Okay. The role of a B cell and its destiny is if it is told to produce a antipathogenic response, what that is an antibody, those Y-shaped little structures. The nomenclature is kind of annoying. A plasma cell is just a fully adult B cell that's secreting these things. Remember the step that we kind of started with today. If a piece of pathogen is given to a B cell by a giant macrophage, that B cell will say, okay, my antibody structure, this little Y right here, happens to bind pretty well to that, meaning that that is going to be like my target right there. If it's given enough go-aheads, it will eventually evolve and differentiate into this plasma cell. The only difference between a naive B cell and a plasma cell is that plasma cell is actually going to secrete antibodies into your bloodstream to fight. A little bit more detail on what an antibody actually looks like. So, antibody is actually a collection of genes in your genome. You can see how there's kind of segments there, right? Gray, dark blue, and blue. Beauty of an antibody is that gray kind of tip up there can fit a lot of different things. Every B cell has a different antibody on its surface. That's the variation that we have in humans to be able to respond to lots of weird looking stuff. So these giant structures are made of protein, like the rest of your genes. What they do essentially, if I wanted to zoom out a little bit, is let's say here's some nasty looking bacteria, right? Kind of floating around, evil. What eventually a plasma cell is going to do, it's going to start secreting these little antibodies out in like the quadrillions of numbers, okay? These are going to go and attach to all these pieces of bacteria. And basically they just kind of flood the thing. Can't move when there's trillions of these things running around you. And they're all binding to just you. This surface right here, this is what takes 3 to 7 days to evolve. Basically, your B cells all have a fight amongst each other to say who's the best one to target this thing that we just saw. There we go, see? Sad face. All right, take a break, but equally kind of run through. What are 3 things you feel okay on? What are 3 things that you're like, I might have to want to ask him about that? Yeah, so we'll reconnect in like 60, 90 seconds, maybe. Okay, so kind of just a relevant story to kind of connect some of this. Obviously, COVID, when you first had it, had never been seen by your body. It had never been seen by any of your ancestors. Most of us had to evolve a response in those 3 to 7 days. Everybody's a little different though. Immunology explains the people that have had it bad, had it kind of little, And those of us that maybe never have had it yet somehow, who else? Nothing yet. The Last of Us. Actually, there's technically a more reasonable explanation for that. It's not as exciting as we think, but so what will happen in a severe COVID or even a non-severe is that pieces of the virus will show up. They will get eaten by macrophages. If they are losing that fight, or if not, they'll still take back the pieces of COVID. They will show the T cells. They'll say, this thing is here, and we are losing this fight, or it's just weird. Part of that response is going to be that a B cell that is very good at binding to that, whatever the spike protein or whatever piece of COVID that you have decided in your immunology, or if your immune system has decided to showcase, it will trigger that evolution into a plasma cell. That plasma cell will secrete, and also all of its clones, like I said, quadrillions of little antibodies that bind perfectly to the COVID virus. Awesome. What that's going to do for us is, number one, like we saw, it can just flood the thing. If you're covered in antibodies, you can't attach into a cell, right? COVID attacks the ACE2 receptor, I think, right, on the lungs. But it's saturated. It can't move. It can't bind to anything. So even though antibodies are a little less wow, wow, go, kill type than T cells, they can do a lot. Equally, remember complement? Remember these hideous little like monsters that find and kill pathogen? Sometimes they just need a little direction. An antibody can find and say, complement, whatever you were doing, destroy this thing that we're on. That is one of the best functions of an antibody, is rallying complement up. Okay, so to meet the other one, met B cells, they do antibodies. T cells have two primary functions. Number one, some T cells will become sort of generals. Those are helper T cells. They coordinate all the other cells and rally them. These will also be called helper or CD4 positive T cells. Again, immunology is one of those fields that is kind of a wash in names and such. Hence why I do want you to have that note card, because I feel as though if it's something you can look up, I'd rather test you on something big. I don't want you to memorize which one is which. I'd rather have that in the notes and then you give me some actual detail. So helper CD4 cell will be rallied just like from the macrophage or the dendritic cell and say, hey, we need help on this thing. It will then spread signals out and say, okay, the immune system needs to get ready. Everybody needs to come out. That's step one. Killer T cells are typically going to be step two in this process. They need direction. Killer T cells are sort of an adult stage T cell rallied by their CD4 cousins to go and kill anything that looks like this piece. So as much press as you're going to get from killer T cells, because they do a lot of work. CD4 cells, these T helper cells, they're really, really, really important. Without them, your B cell function goes down, a lot of your macrophage and like that communication goes down, a lot of bad stuff. So when a CD8 T cell has evolved, it knows its target. It secretes this mass of poisons right here. Lytic mechanisms is a fancy way of explosion mechanisms, way to make a cell explode, right? Now, unlike the B cell, which never actually targets its target, it just secretes tons of antibodies, the T cell is actually making contact and kind of fusing up to it, secreting a bunch of poisons, and then leaving the scene once it's all dead. Now, there is a bit more happening on this slide than just the general overview for the day. Don't worry, promise that we will get to all this. Unlike antibodies, T cells actually form a specialized function on something called a TCR. They actually bind to something called an MHC molecule. We kind of saw that in the beginning. The MHC is sort of a general self cell. In this case, remember, for today, it's just that infected bad cells will display that they are in trouble on that molecule. We'll do the detail next time. Like I said, we're still kind of introducing everybody. So, in that case, how does this happen? How do you evolve a response? This is your intro to that. So, in this case with a T cell, your T cells are constantly, they're kind of stem-like. Remember we've talked about this. Immune cells they have a lot of stem-like qualities. They can change quite a bit. So if you have a big batch of T cells, and remember, this number is more like in the millions of different clones. Each of these has some variation in these little TCR receptors. So what a TCR is a T cell receptor. Think of this kind of like an antibody that it's targeted against something bad. Oh, and actually the image is good enough that can you notice the TCRs are actually all different? Nice. Oh, that was like good. So you have a big pool in your thymus, for example, of T cells that are kind of just sitting there and they can kind of change that protein a little bit. Your genes are a little different in all your T cells. I'll show you that. Eventually, when a macrophage shows up with a piece of bad, it will kind of go to each T cell and say, does this fit? Does this fit? Does this fit? All of these right here did not fit the antigen that was brought forth. What will happen is that only the best one will be chosen, smiling face, and it will kind of like times a million itself. It'll mitosis until cows come home, basically. So the broad concept here is that when you present a piece of unknown pathogen, you present it to a big varied population, and just the one that's the best binder has all the advantages, and it becomes the new pool of T cells right there. And this is sort of like a fun connection to evolution ecology, right? Well, you change the environment and you give an advantage to somebody with a certain niche, their population, their offspring will flourish, and the others, not so much. You don't kill your T cells off every time, they're just sort of ignored. Yeah. A similar thing happens with B cells. The end is just that you don't create like a new cell, you just create a cell that happens to secrete those little antibodies. So again, you're in a process where this piece right here is going to get read. And if that piece is a good fit, then this B cell will be told, okay, make tons of clones of yourself. You have the one that binds the best. You have the variation. And that will make tons of clones of itself. Pretty good. So, like I said, don't worry too much about, this is definitely, you know when props always say like big picture, but then they're lying? I promise I'm not, because I've gotten duped by that so many times. We'll do some, I think over this weekend, I'll try some practice questions, even well, it's a break this weekend. Nobody's gonna watch that. All right, never mind. Next week, after that, I will like prepare some practice questions, because that'll give you more of an idea. So basically what happens is, yeah, this probably should be blue text, sorry, because it's kind of a friendlier repeat, my bad. This should be blue. Basically what happens is, who's ever heard of that shark species that in the womb of the mom shark, they all start coming alive and they start killing each other? And then one comes out that killed all the other ones? Kind of what happens with the B cells. Yeah, it's kind of nasty. Nature's bad. Kind of crazy. Okay. So, cool thing, last thing, like we said, kind of in the beginning. Once you evolve one of these T cells or B cells that is like that finale stage, some of them can stick around. And they will wait until again they see their sort of their mate, that antigen ever again. Now this does bring up a good point with why do people get diseases again then? Disease can also evolve sometimes, or the piece that you have memory against, maybe another piece is a little more stronger in this variant. That can sometimes be the issue. So, actually I have 15 minutes. I wonder if that's like an awkward amount of time. I wonder if this would be perfect to start more like heavy duty material, huh? Yes? Somebody said yes. Yeah, do you have questions, Zach? Yeah.
It was sand tiger sharks, by the way.
Oh, what is that what they're called?
I looked it up.
Okay, good. I just tell the story because it's fun and it's nasty and I hate it. God, this is so gross. Oh, that's okay. Okay. Yeah. So I did also, yeah. So this kind of brings in, now that we're done. So by the way, yeah, actually pat yourselves on the back because that's it for basic players of both times. That's it. B and T cells, adaptive for the most part. All those other little infantrymen before that. So go ahead again. Now's a good time for that 3 and 3. 3 things I'm pretty good at, 3 things that I'm not happy about. This is also kind of ammunition for, people always say like, what do I come to office hours and talk about? Bring those other 3, bring the bad 3. That's what we'll do. Yeah, you can use like next couple minutes, see how people left on your left and your right, see if they have the same stuff or different stuff than you have too. Might be an easy way to consolidate. So as far as like, yeah, kind of questions that I could ask, those of you who had me know the dreaded, one of the easiest clinical format questions will always be, what is not like a similarity between these 2 things? What is not the difference between these 2 things, right? Those of you that have me, give me the thumbs down. Boo me if you don't like those questions, or cheer if you like them.
Ooh.
Yeah, how dare you? They are annoying, they're hard. But that's the format that you see in those med and dental school style exams. That's the way that they can do that. And they don't do open answers the way that we can get some of these done here. Yep, that's right.
So what is not a difference? Just saying like, what is similar?
Yeah, put it in the worst possible way. Remember, I was trained by MDs on how to punish students, right? It's great. But the good thing with ours is that there's a lot more open diagram spacing type stuff, like bigger answers that we can do. Because it's not a, overly massive class. I can grade that, that's okay. Okay, so on to some more fun stuff. It's the last 10 minutes. More interesting stuff, I should say. Equally, I asked you in the beginning, what is the most consequential disease? A lot of you did good. There's a lot of good stuff. I wanted more specificity from some of you, so again, remember, get me those answers more later. Like now, I'm still good about giving you those 2 points back. I just want some like harder, more specific stuff. But who's ever heard the title of a book called Guns, Germs, and Steel? That's a history book, right? Yeah. Germs are going to factor in big. So, first lesson of this story. The immune responses that we have today are sort of littered throughout the animal kingdom. Flies have toll-like receptors. Certain fish have similar receptors in their T cells, but not their B cells, for example. BDJ recombination, which we'll see evolved a little later, onto us. So immune evolution is a very unique way of tracking species in a lot of cases. You sort of see the strengths and weaknesses across the board. Equally, we have absolutely been subject to this. Like that initial slide that I showed, regions where there was livestock were heavy in smallpox. Regions where there was enough water to sustain mosquitoes, or in that case, most peoples from that area are very good against malaria. So over evolutionary time, our immune system is in an arms race constantly against bacteria, viruses, whatever it is. I think this nature photo does decently okay on here. Interestingly, COVID is a unique sort of jump because it sort of was us getting a little too close to a zoonotic disease that maybe we wouldn't have encountered in other scenarios, I guess. And it's also showcased the burden of evolving a new response against something that no ancestry has a very good time against, those corona-like viruses. And as COVID kind of, keeps going, it'll change. There's certain evolutionary pressures that you're seeing on there that will continue to change. And as tired as we all are of the virus, it is very much a changing, if not maybe a living thing, and it'll keep going. Now, big deal is that all your ancestors survived long enough to pass on whatever genes they had in their environment to survive. So in this case, that's why you see a lot of peoples from, like I said, those biocultural evolution areas, you will have specific genes that are very good protection agents against specific pieces. Now, a lot of these are going to be something that we'll meet. Your T cells have a bunch of different variable receptors on them. All kinds of stuff. These are called HLA variants. The more of those you have, the better protected again you are against a random event of infection, because you might have just a ton of different variations. This is also a little bit why, oh yeah, that's a story for another lecture actually. Can't do that one yet. But we have a lot of diseases over here, for example, but there are gene variants that if you have that variation or mutation, you have a protective effect. These are strict mutations as well. These are pretty big deal changes sometimes. Sometimes this is the easy way to pass on. These are a little more strict than some of the other ways we can talk about. So a good example here is, like I brought up, people in the room who have yet to had COVID yet. I know that I was exposed to it because Dr. Minicozy and I were at the locale with one of our close talking faculty, and they were professing on us very closely. Didn't get it. I'm free. And if I survived fall 2020, which had a Monday after Halloween Saturday, I know I didn't have it. Because I know everything was out at that time. So, evolution in humans is it's a very different discussion because we are unique. We did break a lot of rules once fire was a thing. We still very much change in this way, and these variants do protect us. So, is kind of like what I'm talking about. So who's seen the movie World War Z before? Yeah. Do you know that that's actually written about the SARS epidemic? It's an allegory for the whole thing. So when this scene is this guy saying, okay, all the zombies like awoke in this room, boom, but this guy in the light, he was like, this guy just sat there and nothing happened, lucky bastard. That's why variation in population matters. And that's why certain allele combos can defeat certain viruses or bacteria. For example, there are long-term HIV survivors, right? There are certain genetic variants associated with that long-term survivalship. So the variation in the human genome can give you those tools. Now, as far as history shakers and as far as big diseases, you're all down to have your opinions on it. We only had one person say the big one. Malaria. Why do you think malaria kills so many people? It's because it's a single cell parasite, but it hides inside your blood cell. Your immune cells can't kill what they can't find, right? Blood cells are very hard to communicate about what's inside of them. Mosquitoes are also very hard to kill. There's lots of them. When they transport this in, not exactly easy to treat. So, as far as immunity goes, this is one of our biggest examples. Sometimes it's referred to as sickle cell disease, but there is a gene hemoglobin responsible for carrying oxygen in your red blood cells. I think a lot of us have seen this before. If it changes, it has that sickle cell shape in the middle there. That prevents the malaria from hitching a ride. Now, if you have two alleles of this, you have oxygen issues. But as you can tell, the middle is perfect. You have the superpower of being resistant to the most dangerous disease on the planet. This is a less subtle immune change. But for this reason, history played out in a lot of ways. This change right here. All right, so college is about history, heavy-duty shit. You guys ready for this? How we ended up with the consequences in the world today, a lot of stuff. Immunity matters in this case. Okay. So the story, the uncomplicated version of this story is that in the beginning, Genghis Khan is running around here destroying everything, destroying the Middle East, Asia, and Eastern Europe. The only places left without getting hit by Genghis Khan's horde in that like 50-year period, and I'm talking about 20 to 80 million people dead. Like bad world-changing stuff, bad stuff. Here, 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 and here, right? Everywhere else got completely like, the world ended basically. And obviously over here, sorry. Now, Europeans in the West started to get crowded, plague happened. Exploration became the need and the norm. Europeans would go over here, and as some of you talked about, they would bring livestock smallpox, but with their immunity, it would devastate the rest of the Southern and Northern American peoples that were there. So that's one story that a lot of you cited as far as immunity goes, and that did change the course of history. Malaria is the factor here, though. Malaria prevented any colonization there for a long time. That wasn't until in the Peruvian mountains, some of the Spaniards figured out these people in here are not dying. Why? From malaria. Because they had brought malaria over, unfortunately. Peruvians in the mountains had access to a bark of a tree that would prevent them from dying from malaria. We know the substance of this tree today is a quinone, also known as hydroxychloroquinone as well. Quinones were an effective treatment for malaria. Quinones made this happen. All the things that would follow from that. Okay. So what once was a perfect scenario and sort of a shield against what Western Europe was at this time, that collapsed on the finding of quinones, the ability to defeat parasites in this case. Now, in *Sapiens* by Yuval Noah Harari, he talks about a moment here as well, where in this region over here, there's a lot of agriculture and a lot of stuff, they needed a lot of workers. In that time, they would bring slaves from places. They had three options, they would say. Enslave the indigenous population, enslave Eastern Europeans, bring them over, or here, African. That choice in that moment in history be dictated by the fact that in this region, in these regions, the malaria parasite was very prevalent. So when given that choice, who was immune to malaria? Superpower enshrined history and the consequences that we deal with today. So anytime that immunity or germs don't matter, this whole thing is connected around. So I hope that's not too heavy deal of stuff today, but I wanted to show you how powerful some of this stuff is. So meet Wednesday. Give me any questions on the lab today. We'll go from there.